Have you ever wondered how cells, the building blocks of life, communicate with each other? This intriguing world of cell communication is a complex and dynamic dance, an essential process that maintains the harmony of life. Let's start by understanding the local communication between cells. The first method is through gap junctions, tiny channels connecting adjacent cells, allowing them to share ions, water, and small molecules. Imagine them as little tunnels connecting two neighboring houses, enabling the inhabitants to pass notes back and forth. Next, we have contact-dependent signals, a sort of secret handshake between cells. Here, the message-carrying molecule on the surface of one cell physically interacts with the receptor proteins on another cell. It's like whispering a secret message into someone's ear. The third form of local communication is autocrine signals. In this method, cells release signals into the extracellular fluid that influence their own behavior. Think of it as talking to oneself, but for cells. Now, let's shift gears and discuss long-distance communication. The first type is endocrine signaling, where hormones are transported through the bloodstream to distant target cells. It's as if a message in a bottle is sent adrift in the ocean, reaching far-off shores. The second form of long-distance communication is neural signaling. Here, electrical signals travel along nerve cells, triggering the release of neurotransmitters. Picture it as a high-speed train, delivering messages at lightning speed from one station to another. So, whether it's a secret handshake, whispering a message, talking to oneself, sending a message in a bottle, or riding a high-speed train, cells have evolved an array of communication methods to ensure they function in harmony. Now that we know how cells communicate, let's delve deeper into the fascinating world of ligand binding. This dance of molecules is another integral part of the cell communication story, which we will explore in the next scene. So, stay tuned. Imagine a ballroom dance where ligands are the dancers and receptors are their partners. The music starts to play, and our lipophilic ligand, the dancer with a love for fats, makes its way across the dance floor. It travels through the phospholipid barrier of the cell, gliding effortlessly as it's drawn towards the nucleus, the heart of the cell. Once there, it meets its partner, the intracellular receptor, and they form a complex, just like two dancers coming together. This complex then binds to specific regions of DNA, triggering the production of proteins and setting off a cascade of cellular events. That's the dance of the lipophilic ligand, a slow, graceful waltz that happens inside the cell. Now, let's turn our attention to the lipophobic ligand. This dancer isn't a fan of fats, so it can't cross the cell membrane to join the dance inside. Instead, it does a lively quick step on the cell surface. It binds to a cell surface receptor which acts like a switch, turning on a signal transduction pathway. This pathway is like a line of dancers, each passing on the message to the next, amplifying the signal until it reaches the intended cellular machinery. The result? A response is triggered in the cell, whether it's a change in activity, a movement, or the initiation of a process. So whether it's the slow, graceful waltz of the lipophilic ligand inside the cell, or the lively quickstep of the lipophobic ligand on the cell surface, each ligand has its own dance. Each dance triggers a sequence of events that leads to a response within the cell. Just as dancers have different styles, so too do cell surface receptors. Let's explore these types next. Like a key fitting into a lock, ligands bind to specific receptors. But did you know there are 4 major groups of these receptors? Let's dive in. First up, we have ion channel-linked receptors. These are like gates that open or close in response to a ligand, allowing ions to pass through the cell membrane. Then, there are G-protein-linked receptors. These act like molecular switches, turning on or off in response to a ligand to activate other proteins. Thirdly, we have enzyme-linked receptors. These receptors have built-in enzymes that get activated when a ligand binds. The last group is the intracellular receptors, which are located inside the cell, and they respond to lipophilic ligands that can cross the cell membrane. Each group has a unique way of responding to ligands, which adds to the complexity and diversity of cell communication. These receptors play a critical role in signal transduction, a process that we will discuss next. Ever wondered how a small signal can lead to a big response in a cell? That's where cascades and signal amplification come in. Signal transduction is the process by which a cell converts one kind of signal or stimulus into another. This process often involves cascades, a series of biochemical reactions where the product of one reaction triggers the next. It's like a biological domino effect. Signal amplification, on the other hand, is where the impact of a signal is increased. Think of it as turning up the volume on a song. The original sound is the same, but the impact is much greater. In cells, this is achieved by activating multiple copies of the next enzyme in the cascade at each step of the process. Together, cascades and signal amplification ensure that even the smallest signal can have a significant effect. It's like shouting into a canyon and hearing your voice echo back 100 times stronger. This is critical in biology, where sometimes the signal might be a single molecule. One key player in this process is calcium, an intracellular messenger with a number of roles. Calcium ions can directly regulate many proteins, altering their structure and function. They can also bind to specific proteins like calmodulin to change their function. Calcium ions can even act as a second messenger in many hormone and neurotransmitter pathways, helping to carry the signal inside the cell. Calcium can also trigger the release of other intracellular messengers, amplifying the original signal. Lastly, calcium ions can create an electric charge difference across the cell membrane, influencing the activity of voltage-gated ion channels and the flow of other ions into and out of the cell. Together, these actions of calcium help to amplify and spread the signal throughout the cell, ensuring a robust response to even the smallest of stimuli. But it's not all about the signal. Sometimes the absence of a signal is just as important. Let's explore this next. In the world of cell communication, it's not just about sending and receiving messages. It's also about how these messages are interpreted. Let's dive into the modulation of signal pathways. First, let's talk about specificity. Each receptor is designed to respond to a specific ligand, much like a lock and key mechanism. This ensures that each signal is received by the right receptor and triggers the appropriate response. Next, competition. Sometimes different ligands compete for the same receptor. Imagine this as a game of musical chairs, where the ligands are the players and the receptor is the chair. The ligand that binds to the receptor first gets to deliver its message. Affinity relates to the strength of the bond between the receptor and its ligand. The stronger the bond, the more likely the ligand is to remain attached to the receptor, and the more potent its effect. Think of it as a handshake. The firmer the grip, the more impactful the interaction. Saturation refers to the point at which all receptors are occupied by ligands. Once a receptor is saturated, additional ligands cannot bind, limiting the intensity of the response. Now let's explore the role of upregulation and downregulation in modulating cell responses. Upregulation is like turning up the volume on a radio. The cell increases the number of receptors, amplifying the response to a specific ligand. Downregulation, on the other hand, is like turning down the volume. The cell decreases the number of receptors, dampening the response. Finally, pathway termination is the process by which the cell ends a signal. Think of it as hanging up the phone after a call. It's an essential step that prevents overstimulation and maintains balance within the cell. Now that we've explored the intricate world of cell communication, remember that this is just the tip of the iceberg. There's a whole universe inside each cell, waiting to be discovered. So, what have we learned about cell communication today? Quite a lot, it seems. We've dived into the fascinating realm of local and long-distance communication, understanding the dynamics of signal pathways. We've unraveled the mystery behind lipophilic and lipophobic ligand binding, revealing the intricate sequences of events that follow. We've explored the diverse world of cell surface receptors, identifying 4 major groups, each with a unique role in the communication process. We've dissected the role of cascades and signal amplification in signal transduction and how they amplify the messages being sent. We've discovered the novel signal molecules like calcium and how they act as intracellular messengers. We've also touched on the modulation of signal pathways and the concepts of specificity, competition, affinity, and saturation. And finally, we've compared neural and endocrine reflexes and explored complex reflex pathways. Remember, every moment of every day, your cells are communicating. They're telling a story of life that we're only beginning to understand.